Hello students, let us discuss about the brain. Let us study the anatomy and the physiology of the brain. In the previous class, we have studied about the spinal cord and the spinal nerves. So from today's class onwards, let us study about the brain. So we all know brain is the controlling center of the body. It is very important. Without brain, a person cannot make a decision or he cannot control the homeostasis of the body. So brain is very important for contributing and controlling the homeostasis in the body, both externally and internally, by receiving sensory input, integrating new and stored information, making decisions, and causing motor activities. So the control or the order is given by the brain. So it is acting as an integrating center where it can process the information, store the information, and make decisions and lead to all the physiological functions, that is both somatic as well as the autonomic functions of the body. The brain is the control center for registering sensations, feelings, emotions, and other things, correlating them with one another and with the stored information, making decisions and taking actions. That is what we call it as the sixth sense. So this is the brain. And you can say, the memory, the to maintain the relationship with the person to person, and the emotions, everything is controlled by the brain. It is also the center for the intellect, emotions, behavior, and memory, as I told you. It directs our behavior towards others. So, by controlling the brain activity and by training, we can a child acquires many qualities throughout the life. The brain is the portion of the central nervous system. So, central nervous system includes the brain and the spinal cord. It is present inside the cranium. So, it is protected by the skull called the cranium. So, there are about 100 billion neurons and 10 to 50 trillion neuroglia which make up the brain. And what is the weight of the brain? It is around 1300 grams in the adult. So, even though it is very small in size, it is very important for the control of the various physiological mechanisms that is happening in the body. So, on average, each neuron forms 1000 synapses with the other neurons. So, the total number of synapses, about 1000 trillion or 10 power 15, is larger than the number of stars in the galaxy. So, imagine the brain can even function better, faster than the computer. So, that is the beauty and the amaze of the brain. The brain, along with the spinal cord and the 12 pairs of cranial nerves, connect with the brain to form the control center of the human body. That is why it is called as the central nervous system. Let us have a look about the organization of the brain, how it is protected, and the blood supply. First, let us discuss about the development of the brain. So, how the brain is developed from the small fertilized ovum, and how it is developed into a brain. So, what are the structures which make up the different parts of the brain? So, the brain and the spinal cord develop from the ectoderm arranged in tubular structure called the neural tube. So, when you see the structure, you can see a neural tube, central neural tube, and an ectoderm. So, the brain and the spinal cord develop from the ectoderm, which is arranged in a tubular structure, which is called as the neural tube. The anterior part of the neural tube expands and constrictions appear that create three regions called the primary brain vesicles. So, as the fetus develops, passes through many stages, that is the first month, the second month, third month, sixth month, and so on, the different parts of the brain develop. So, the folic acid is very important for the brain development. The initial structures that develop in a fetus is the brain. So, it is very important that the pregnant mother consumes folic acid along with other calcium and the vitamins. So, the brain development is very, very important. So, the anterior part of the neural tube forms three regions called as the proencephalon, mesencephalon, and rhombencephalon. Proencephalon forms the forebrain, mesencephalon forms the midbrain, and rhombencephalon forms the hindbrain. The walls of these brain develop into nervous tissue of the brain, while the space in the tube will be forming ventricles or spaces within the brain called as ventricles. So, what are the different structures and what give rise to the different parts of the brain? The mesencephalon give rise to the midbrain and aqueduct of the midbrain, cerebral aqueduct. Both the prosencephalon and the rhombencephalon will further divide, forming secondary brain vesicles. The prosencephalon give rise to the telencephalon and diencephalon, and the rhombencephalon develops into the metencephalon and the myelencephalon. The cerebrum and the lateral ventricles are developed from the telencephalon, whereas the diencephalon forms the thalamus, hypothalamus, and the epithalamus. So, all these names you would be familiar with by now. The metencephalon becomes the pons, cerebellum, and the upper part of the fourth ventricle, whereas the myelencephalon forms the medulla oblongata and lower part of the fourth ventricle. I think from this picture you can very well understand how the development of the brain takes place. You can see the first picture. There is a 3 to 4 week embryo. So what happens? There is a wall and a cavity. So as I told you, the ectoderm and the cavity. So there is the brain and the spinal cord developed from the ectoderm neural tube. So the wall forms the various structures: cerebrum, cerebellum, medulla oblongata, etc. Whereas the cavity forms the vesicle. So here you can see three different structures: prosencephalon, mesencephalon, and rhombencephalon, which will in future they develop into forebrain, midbrain, and hindbrain respectively. The prosencephalon will further divide. You can see the division here. This is happening in the 5 week embryo. See, this is less than a month. So 5 week embryo, before you could even realize that a female is pregnant. Okay, all this will happen. So that is why the doctors say you should not take any medicines during pregnancy. That is very very important, because it will immediately have a reaction on the fetus, developing fetus, and it will result in the impairment of the brain development. So that is why it will lead to many complications when the child is born later. That is, he or she can develop into a special child. So that is very important. So the prosencephalon is further divided into telencephalon, which will further divided into cerebrum and the lateral ventricles. The cavity is called the lateral ventricles. Whereas the diencephalon, which is formed from the prosencephalon, will form the thalamus, hypothalamus, and the epithalamus. These are the endocrine system: thalamus, hypothalamus, and epithalamus, which will further at a 5 week, when it grows bigger, it will form the thalamus, hypothalamus, and the epithalamus, and the third ventricles. So these are the walls and these are the cavities. The mesencephalon does not undergo any change. It directly develops into midbrain, which is seen in the yellow colored structure and the aqueduct of the midbrain. Whereas the rhombencephalon, which is the hind part of the brain, is divided into metencephalon and myelencephalon. The metencephalon forms the pons, cerebellum, and the upper part of fourth ventricle, whereas the myelencephalon forms the medulla oblongata and lower part of the fourth ventricle. So this is how the development of the brain takes place. So in this structure, in this picture, you can see the origin of the nervous system. That is a dorsal view of an embryo in which the neural folds have partially united, forming the early neural tube. You can see this is the neural tube. That is the dorsal view of the neural tube, head and the tail end. And you can see the neural tube in the center. There are many grooves: neural groove, neural folds, and neural plate. So which will further develop. The nervous system begins developing in the 3rd week from a thickening of ectoderm called the neural plate. You can see the ectoderm here, endoderm, okay, which forms the neural plate. Here you can see the neural plate. See how the further development takes place. The ectoderm, neural fold. See, it is not undergoing any folding here, whereas here it undergoes some kind of folding. And this is the ectoderm forms the brain. See how closure and the structure difference takes place. So here you can see very clearly what happens. The mesencephalon does not undergo any change. Mesencephalon forms the midbrain. Here you can see this is the midbrain. This region there is no change. Whereas in the forebrain you can see the prosencephalon. This divides into mesencephalon and the diencephalon and the telencephalon. Diencephalon and the telencephalon. The telencephalon will give rise to the cerebrum. You can see the telencephalon will give rise to the initial part of the brain called as the cerebrum, see cerebral hemisphere, and the diencephalon give rise to the thalamus, hypothalamus, and the epithalamus, and also the third ventricle. So that is what is the role of the forebrain. That is the prosencephalon divides into telencephalon and diencephalon. Telencephalon give rise to the cerebrum, and diencephalon give rise to the thalamus, hypothalamus, epithalamus, and the third ventricle. So this is what is happening. Now let us look at the hind brain. So here you can see there is a rhombencephalon which forms the hind brain, is divided into metencephalon and the myelencephalon. So this metencephalon and the myelencephalon further is forms the that is, you can see the metencephalon forms the cerebrum and the pons. See, these are two regions, one this side and one that side. The metencephalon forms the cerebellum and the pons, whereas the myelencephalon forms the medulla oblongata. So this is the structure. So this you can see the brainstem. Here it forms a pons, medulla oblongata, spinal cord. These forms the brainstem. The upper part is made up of cerebral hemisphere, diencephalon, which is the hypothalamus, thalamus, and the epithalamus, and this is the cerebellum, and these are the brainstem. Okay, now let us see the major parts of the brain. So as we have seen in this picture, the adult brain consists of four major parts: the brainstem, the cerebellum, diencephalon, and the cerebrum. So what are the four parts? The brainstem, cerebellum, diencephalon, and the cerebrum. So the brainstem is continuous with the spinal cord, that is the medulla oblongata, and consists of medulla oblongata, pons, and midbrain. So this forms the brainstem. So this is continuous with the spinal cord, and it consists of medulla oblongata, pons, and the midbrain. Posterior to the brainstem is the cerebellum. Cerebellum, it is called as the little brain. Superior, upper side of the brainstem is the diencephalon, which consists of the thalamus, hypothalamus, and the epithalamus, the endocrine centers of the brain. So supported on the diencephalon on the brainstem is the cerebrum, which is the largest part of the brain. So these are the different structures or parts of the brain. What are they? The brainstem, cerebellum, diencephalon, and cerebrum. The brainstem is formed by the medulla oblongata, pons, and midbrain. The posterior, that is above that you can see the cerebellum, and above the cerebellum you can see the diencephalon, which forms the thalamus, hypothalamus, and the epithalamus. On top of this you can see the cerebrum, which is the largest part of the brain. So here you can see the cerebrum, the diencephalon, which forms the thalamus, hypothalamus, and the epithalamus. So these three are the structures of the diencephalon. Then you can see the brainstem, which is made up of the midbrain, pons, medulla oblongata, and this is the cerebellum. This is the spinal cord. So this is the structure of the brain. So there is another structure which is given, the same thing, the cerebrum, diencephalon containing thalamus, hypothalamus, the brainstem containing the midbrain, pons, and medulla oblongata, and the spinal cord. This is the cerebellum. By now I think you would have known the structure of the brain very clearly. Now let us see the protective coverings of the brain. So just like the spinal cord, the brain is also protected by bony structure called as the cranium. So cranium, which is called as the skull, and the cranial meninges, just like the spinal cord, which we have studied in the earlier classes. This brain is also protected by meninges called as the cranial meninges. The cranial meninges are continuous with the spinal meninges and have the same basic structure, containing the outer dura mater, middle arachnoid matter, and the inner pia mater. So this is continuous. So in the previous chapter also we studied that the meninges are continuous. That is, the cranial meninges and the spinal meninges are continuous. The cranial dura mater has two layers. The spinal dura mater has only one layer. So that is the difference. The cranial dura mater has two layers because it has to protect the very soft structure, that is the brain. Whereas the spinal dura mater has only one layer. The two dural layers around the brain are fused together, and there is also no epidural space around the brain. You can see the blood vessels entering the brain tissue. They pass along the surface of the brain and penetrate inwards. You can see the blood vessels. You can very well see the brain of a goat. It is very much similar to the goat. When you take this brain of the goat, the upper structure is surrounded by the blood vessels, which you can see very clearly, which passes through the fissures and the ulcers. So the blood vessel enter the brain, passes along the surface of the brain and penetrate inwards. They are sheathed by loose fitting sleeve of pia matter. So this is how the brain is being protected by the outer cranium, which is the skull. There is a bony structure called as the skull, the cranial meninges, and the supply of blood vessels. So here this picture shows the covering cranial bones and the cranial meninges that protect the bone. This is the skin, and this is the bone of the cranium. And here you can see the meninges, the dura mater, outer dura mater, which is a double layered structure but still connected together, the middle arachnoid matter, and the pia mater. So these are the structures, and you can see the blood vessels. So how the blood vessels are passing through the surface of the brain. So the brain is protected inside. These are the cerebral cortex, and you can see the supraarachnoid space here. Okay, now let us see the extensions of the dura mater. So there are three extensions of the dura mater which separate the brain parts. So you know that there are two cerebellum, two cerebrum. Okay, the cerebrum, the right side of the cerebrum is called as the right hemisphere, and left side of the brain is called as the left hemisphere. So there are two hemispheres which are separated by a center, okay, called as the falsi or a separation. Okay, so similarly the cerebellum is also the left side and the right side. It is divided into left and the right cerebellum, and the cerebrum is separated from the cerebellum. So this is separated by extensions of the dura mater called as the falx cerebri. The falx cerebri separates the two hemispheres of the cerebrum, right and the left cerebrum. The falx cerebelli separates the two hemispheres of the cerebellum. From the name itself, we can understand. There is a third one called as the Tentorium Cerebelli, which separates the cerebrum from the cerebellum, anterior and the posterior regions of the brain. So here you can see the dura mater, the falx cerebri. Cerebri separates the right and left hemisphere of the cerebrum. You can see the frontal bone, and the second one is the falx cerebelli, which separates the right and left side of the cerebellum. Tentorium cerebelli separates the cerebrum from the cerebellum. So these are the three structures. Let us see the brain blood flow and the blood brain barrier. So the blood flows to the brain. Brain cannot function. The brain has to be supplied with the blood vessel and fuel. That is, fuel is given by the glucose. If the brain does not get enough fuel in the form of glucose, immediately you can experience the fatigue of the brain, and it will result in fainting or collapsing. So the blood flow and the blood sugar level is very, very important. That is why when you are hungry, when there is a low concentration of sugar in the blood, immediately you will feel fainting. You will feel like fainting, and that is you can tell in Tamil, pasimayakam. You can say pasimayakam. It is immediately reflected in the brain. So the blood flows to the brain mainly via the internal carotid and the vertebral arteries. So these are the arteries that supply the blood to the brain. What is it? It is the internal carotid artery and the vertebral artery. The internal jugular veins return blood from the head to the heart. So the veins are the jugular. You know, jugular veins are present in the neck region. So the jugular veins are very, very important. So this is the life saying. If the jugular vein is cut by biting or something, immediately the person dies. You know, when you take when you want to inject IV or any injection, you take the hand and you will insert the needle in the hand. When the during I mean, what do you say? If you don't get the nerves, immediately the final thing, when a person is fainting or collapsing or not in a stage where the veins are visible, they will inject the IV through the jugular veins. So jugular veins are running, the in the neck region, sides of the neck. So this is very important. So blood flow to the brain is through the internal carotid artery and the vertebral arteries, whereas the outflow of the blood is from the internal jugular veins. So, in an adult, the brain represents only 2% of the total body weight. It is very, very small, but consumes 20% of oxygen and glucose even when it is at rest. So the brain is continuously working. A man cannot I mean be without thinking. Okay, the brain is always active. Even when he is sleeping, the brain is active. Even when he is at rest, the brain is very active. So this is though, even though the brain is very smaller in size, it consumes 20% of oxygen and also glucose. So neurons synthesize ATP almost exclusively from glucose. So glucose is the only fuel. The second kind of fuel is the ketone bodies. So they use the glucose and the oxygen, and the oxidation of glucose will give rise to the ATP molecules for the brain function. When the activity of the neurons and neuroglia increases in the region of the brain, then the blood flow to that area also increases. You can suddenly feel some rush of the blood inside the brain. Okay, so when the particular area of the brain, the activity of the neurons and neuroglia are increased, so the blood flow also increases to the region. So even a brief slowing of the brain blood flow may cause unconsciousness. So when there is an injury, or when the blood flow becomes less immediately, the person may become unconscious. That is what is happening during brain death. So what happens if he goes to unconscious state and coma? So that is what that may be because of a stroke or a block or a clot or a hemorrhage. So whatever it may be, when there is a blood supply is cut, okay, he may fall unconscious. So similarly, if there is an interruption in the blood flow for even 1 or 2 minutes, it impairs the neuronal function. So that is what happens during the childbirth. So during the childbirth, what happens when in a normal delivery? What happens? The normal delivery is of many types. It can be born naturally, or we can use some instruments like the forceps to bring the baby, to pull out the baby. So when you can see the forceps baby, the head will be elongated. So during these conditions, or sometimes the baby, if it stays inside the inside for a long period of time, it can consume meconium. So all this will result in the damage of the brain, and what will happen? The baby will be born with some defect, okay, brain defect, or some slow learners. That is, you can call as a special child. So even there is an impairment of blood flow for 1 or 2 minutes, the neuronal function is affected, and the total deprivation of oxygen for about 4 minutes causes permanent injury. If there is no oxygen supplied to the brain for 4 minutes, also it will cause serious damage to the brain. No glucose is stored in the brain. So glucose, no, the brain does not contain any storage tissue. Okay, the glucose has to be continuously supplied through the blood flow. So because glucose cannot be stored in the brain, it has to be supplied continuously. So if the blood entering the brain contains less amount of glucose, then I told you, maybe because of starvation, or maybe because of any other condition, it will result in dizziness. So mental confusion. If it doesn't eat for a long time, hunger, it will result in mental confusion, convulsions, seizures, and loss of consciousness also can happen. What is blood-brain barrier? So blood-brain barrier is very, very important. It will protect the brain from harmful substances and pathogens by preventing a passage of many substances from the blood into the brain tissue. So when the blood is flowing into the brain, it has to be monitored. So it has to go through the blood-brain barrier, which acts as a filter and prevents the entry of the pathogens and other toxic substances into the brain. So what is blood-brain barrier? It is mainly tight junctions of the brain, which will seal the endothelial cells of the brain capillaries. So along with the thick basement membrane around the capillaries. So this is what is the blood-brain barrier. It is made up of tight junctions which seals the endothelial cells of the brain capillaries along with the thick basement membrane around the capillaries. So we know that is the astrocytes. Astrocytes, one type of neuroglia. They press up against the capillaries and secrete chemicals. I think you would have studied about the astrocytes. There is a neuroglial cells. They secrete chemicals that maintain the permeability characteristics of the tight junction. So they only decide what should go inside and what should not go inside. The transport permeability is maintained by the tight junctions by the secretion of these chemicals. Whereas a few water soluble substances such as glucose cross the blood brain barrier by active transport. Substances like creatinine, urea, and ions cross the blood brain barrier very slowly. Whereas the other bigger substances like the proteins, antibiotics, okay, larger compounds do not pass from the blood into the brain tissue. Whereas you can see the protein drugs and all passes through the liver very easily. Okay, that is why liver damage happens very quickly. Whereas the brain does not allow the passage of bigger molecules, proteins and antibiotics to pass into the brain. That is why the blood brain barrier acts as a protective layer. Whereas lipid soluble substances such as oxygen, carbon dioxide, alcohol. So what happens now when you consume alcohol? Immediately you will feel all the, you know, you feel the dizzy, you will feel the pleasure, you will feel, intoxicated, whatever you call it, addiction, the addiction center of the brain. That is because alcohol can easily cross the blood brain barrier, and the anesthetic agents also can cross the blood brain barrier. So lipid soluble substances can easily cross the blood brain barrier. When there is trauma or injury or toxins and inflammation, these will result in the breakdown of the blood brain barrier. Trauma, any injury to the head, to the brain, or any brain fever. So these, any inflammation caused by falling or something, will definitely damage the skull and the internal parts of the body, and it will also break down the blood brain barrier. So that is how the brain is being protected. So this is the structure of the brain, the outline of the brain. So in the next class, we will discuss about the cerebrospinal fluid. So cerebrospinal fluid is also a protective fluid. Apart from the skull and the meninges, cerebrospinal fluid also protects the brain by acting as a shock absorber, insulator, which flows into the ventricles. So we will study about this in the next class.